Students, this is chapter number 11, Biotechnology, Principles and Processes. And in this chapter, this is video number 5. And in this video, if you have not seen the previous videos, please go through them. In the previous video, we were discussing about the process of recombinant DNA technology. And in that, we had already discussed about the two videos, the two processes, that is isolation of the genetic material and cutting of the DNA at specific location. And in this video, I'll be explaining the rest of the 4 steps. So let's start with amplification of gene of interest using PCR. So let us understand what is a PCR machine. Its full form is polymerase chain reaction. And it is a machine where we synthesize multiple copies of gene of interest in vitro using 2 sets of primer and enzyme known as DNA polymerase. What are primers? Primers are small chemically synthesized oligonucleotides that are complementary to the region of the DNA. What are the steps of PCR? The steps are first denaturation, second annealing, third extension. Now, let's start with denaturation. What is denaturation? It is the heating of the target DNA gene of interest, that is our gene of interest, at a high temperature of 94 degrees Celsius to separate the strands. Each strand in this acts as a template to synthesize DNA. Next is annealing. What is annealing? It is joining of the 2 primers at nearly 52 degrees Celsius at the 3rd carbon end of the DNA templates. You can see over here, the primer is being joined at the 3rd carbon end of the DNA template. Now, the next step is extension. Extension is the addition of the nucleotide to the primer. Using a DNA polymerase, and in this case, we use Taq polymerase. Taq polymerase is an enzyme which is excreted out, isolated from the bacteria Thermus aquaticus. This polymerase can withstand high temperature. It remains active at high temperature. And we all know that during denaturation, we have given a very high temperature to separate the DNA. Now, through continuous replication, the DNA segment is amplified to nearly 1 billion copies. The amplified fragment can be used to ligate a vector for further cloning. Now, insertion of recombinant DNA into the host cell, the next step. This we have studied in video 2 and video 3 in detail, where we have seen that there are several methods of introducing the ligated DNA into the recipient cell. And how do we make it? By heat shock treatment, the bacteria receives the DNA, then there is microinjection, then there is ballistic gene gun. Now the recipient cell, after becoming competent, receives the recombinant DNA. If the recombinant DNA is bearing ampicillin resistant gene, is transferred into the E. coli, the host becomes ampicillin resistant cell. And if the transformed cell are spread over agar plate containing ampicillin, only the transformant will grow and the non-transformant will not grow. This also in detail has been explained in video 3. Under cloning vector. So ultimately, what is our goal? Our goal is to obtain the foreign gene product, and the foreign gene product, the recombinant DNA, produces the desirable protein. If the protein encoding foreign gene is expressed in a heterologous host, then the protein is known as the recombinant protein. Now, how do we obtain it? There are different methods of obtaining this product. Now, the cells with the foreign genes are being grown in the lab, in the laboratory, and the culture are used to extract the desired protein and purify it using separation technique. The very first separation technique is the continuous culture system. Here, the used medium is drained out from one side while fresh medium is added from the other side. It maintains the cell more physiologically active, and so it produces large biomass and yields the desired protein. Next is the bioreactor. There are two types of bioreactor. Next is the bioreactor. Let us understand what is a bioreactor. Bioreactors are vessels in which the raw materials are biologically converted into specific product, specific enzymes, using microbial, plant, animal, or human cell. Bioreactors are used to produce large quantity of products. They can process 100 to 1,000 liters of culture. The bioreactor provides optimal growth conditions like temperature, pH, substrate, salt, vitamin, oxygen, and get the desired product. Now, there are two types of bioreactor you can see over here. One is a simple stirred tank bioreactor, and the second is the sparge seared tank bioreactor. Now, let us see about the stirred tank reactor. That is the simple one. It is the commonly used, and it is usually cylindrical or with a curved base. Now, this curved base facilitates the mixing of the reactor contents. The stirrer facilitates even mixing of and oxygen availability. Alternatively, air is bubbled through the reactor. What are the different parts of the bioreactor? You will find an agitator system, an oxygen delivery system, a foam control system, a temperature control system, a pH control system, and sampling ports for periodic withdrawal of the culture. Now, after the product has been produced, then the next step is downstreaming. What is downstreaming? It is a series of processes where you separate the product, purify the product after the biosynthetic stage, and then you give it to the send it to the market. So basically, downstreaming is a series of processes where separation, purification is done. Then the product is, if required, then it is formulated with suitable preservatives. Such formulation then undergoes clinical trials and strict quality control testing. And then after that, it is sent to the market. Thank you. And I hope that you all have understood this chapter.